My name is Adheesh Chincholi, and today I'll be presenting my project titled Computational Analysis of Aptamer-Based Origami Targeting the EpCAM Receptor in Glioma Cells. In this presentation, I'll explain how computational tools can be used to design aptamer-based nanostructures that specifically target cancer cells expressing the EpCAM receptor. My research combines bioinformatics, molecular modeling, and nanotechnology to explore a potential pathway for precise cancer therapy. Glioma is a type of tumor that arises from the glial cells of the nervous system. It's one of the most aggressive and deadly forms of brain cancer. In this study, I focused on the EpCAM receptor, a cell surface protein that is highly overexpressed on glioma cells' surface, but not in normal cells. This makes it an ideal biomarker for targeted therapies. To design molecules that can bind to this receptor, I used aptamers, which are short nucleic acid sequences that can recognize and bind specific proteins with high accuracy. I computationally modeled these aptamers and docked them onto the 3D structure of EpCAM, which I predicted using AlphaFold 3. The results showed strong binding interactions, suggesting that these aptamer-EpCAM complexes could potentially be used for detection or targeted treatment of gliomas. Now, gliomas vary greatly depending on the grade and aggressiveness. For example, in grade 4 glioma, also known as glioblastoma multiforme, the life expectancy of a patient is only 12 to 15 months, even with treatment. This highlights the urgent need for more effective therapies that can precisely target tumor cells while minimizing harm to healthy tissue. One of the defining characteristics of glioma cells is the overexpression of the EpCAM receptor. Overexpression simply means that these cells have a much higher number of these receptors on their surfaces than normal cells. This property can be exploited to differentiate cancer cells from healthy cells, making EpCAM a valuable biomarker for targeted therapy. Now, glioma originates in the brain or spinal cord, and more commonly, it originates in the glial cells of the brain, which is responsible for supporting and protecting neurons. The symptoms of glioma, like headaches, seizures, blurry vision, or changes in cognition, often appear late in the disease, which is one reason for its high mortality rate. Current treatments rely on surgery, radiation, and chemotherapy, which are costly, lengthy, and often come with severe side effects because they lack precision. By introducing a targeted molecular approach like the one I'm presenting today, I can aim to improve the precision and reduce the toxicity of glioma treatments. Now, my objective was to computationally model how aptamers can bind to the EpCAM receptor and to design a DNA origami capable of delivering therapeutic molecules specifically to glioma cells. My hypothesis was that by designing and analyzing these aptamer EpCAM complexes in silico or through computational simulations, I could create a highly selective and stable drug delivery vehicle that minimizes side effects and maximizes treatment precision. Now, my methodology included four main stages. First, I used AlphaFold 3, an advanced AI-based protein protection prediction tool, to generate the 3D structure of the EpCAM receptor based on its amino acid sequence. This gave me an accurate model. I selected known EpCAM binding aptamers secondly from literature and performed molecular docking simulations to study their interactions. This allowed me to identify which aptamers form the most stable complexes, focusing on hydrogen bonds and salt bridges that indicate strong binding. Third, using CAD Nano, I designed rectangular and bundle-shaped DNA origami scaffolds. The aptamer was integrated onto one face of the scaffold, acting as the targeting element. Finally, I modeled a drug delivery system that carried levodopa, an anti-cancer drug, stabilized with silk for controlled release and mechanical stability. This served as a proof of concept for targeted therapy. Now, this slide summarizes how the drug delivery system would work. The aptamer recognizes and binds to the EpCAM receptor on the glioma cell surface. The DNA origami structure carries the anti-cancer drug, and the binding occurs. The drug can be released directly into the cancer cell. This targeted approach minimizes damage to healthy brain cells and represents a step towards personalized nanomedicine. Now, my docking analysis showed that the A2 and A2M2 aptamers, which I had named previously, had the strongest and most stable interactions with EpCAM. In total, about 20 stable interactions were observed in the A2M2 complex, suggesting high binding affinity and selectivity. The simulation confirmed that the design DNA origami could successfully accommodate the aptamer drug silk complex while maintaining its structural integrity. The graph here shows the number of hydrogen bonds and salt bridges for each aptamer, with A2 and A2M2 standing out as the top performers. To understand how genetic variability might affect binding with EpCAM, I used the variant effect predictor tool to analyze mutations in the EpCAM receptor found in cancer patients. One significant mutation is called THR115MET. This missense mutation changes one amino acid in the receptor, potentially altering how the aptamer binds. Such insights are crucial for designing aptamers that remain effective across different patients, ensuring that therapies are adaptable to real-world genetic diversity. In theory, aptamer-anchored DNA origami could locate and bind to glioma cells, expressing EpCAM with high precision. This would allow the therapeutic payload, such as a drug, RNA molecule, or other treatment agents, to be released directly into the tumor cells. With the integration of aptamers with DNA origami, it opens up a promising avenue for low toxicity, cost-effective, and highly targeted cancer therapies. While this work was conducted computationally, my next step would be experimental validation to test these interactions in vitro and in vivo. In conclusion, the study demonstrates how computational modeling can accelerate the design of targeted nanotherapeutics. By combining aptamers, DNA origami, and AI-based modeling tools like AlphaFold, we can move closer to personalized and precise treatments for aggressive cancers such as glioma. Thank you so much for listening.